Michaela Ortega Donahoe and Khalil Robinson. This project helps high school teachers spark student interest in biotechnology across the greater Los Angeles region. Thank you, Aron and Michaela and Khalil.
All right, thank you for the introduction. Am I sharing the correct screen? Yeah, I think so. Hi, everyone. My name is Aron Kamajaya. I am very happy to kick off this ATE, the 2026 ATE project talk, I guess, and tell you about our project. Again, I'm Aron Kamajaya. I'm the program director for the biotechnology program at Los Angeles Pierce College in the sunny Southern California. And today, we actually have two active ATE projects, and one of them is sunsetting this year. In this presentation, I'm going to tell you about the second ATE project, which is the IGD-LA, stand for Illumina Genomic Discovery, Los Angeles. So this Illumina Genomic Discovery essentially have two major objective that somewhat intertwined to one another. The first one is to raise the champion leaders in biotech that can introduce students who are primarily going, high school students who are primarily going to the career in patient care into the world of biotechnology, exposing them to career opportunity available to them in the region, and also the academic path that can help them to get there, not only at Pierce College, but also the training program in the area. The second objective is to expand access to this new technology, next generation sequencing DNA technology, to bioscience classroom, both at community college and high school classroom. So and today, this is our first presentation, as you can see from the title, Illumina Genomic Discovery #1. In this presentation, I'm going to give an outline on how this project built upon the previous project. And then here I also bring a student of mine, Khalil Robinson. Khalil, do you want to say hi? He's, okay, camera may be off, but anyway. So I'll bring him to and he will gonna talk about his project to illustrate how we utilize the next generation DNA sequencing technology to train the biotech workforce through innovative, authentic projects. So without further ado, so first I'm going to describe a little bit about our program at Los Angeles Pierce College, which was catalyzed. The development was actually catalyzed by the first ATE project that we have. So if you have listened to my talk previously, you probably know me as the Taq polymerase guy. So if you're still wondering, again, the first project is meant to build the innovative student internship revolving around the Taq polymerase biomanufacturing. So if you want to incorporate biomanufacturing on Taq polymerase, which the structure is displayed behind in the background, feel free to hit me up. I mean, the curriculum is available through Carolina, but if you ask me or Dave, who's also here from Cold Spring Harbor, we'll be more than happy to provide support and perhaps share resources. So from there, our biotechnology program initially started out with only the research path. It has a stackable certificate, level 1, which was not displayed here, 13 unit, can build into the 20 units level 2 certificate, and that can get into associate degree. And then since then, we built the biomanufacturing, not the research part, but the manufacturing complementary pathway side of the biotech, and then both lead to associate degree. And together this year, we actually launching our baccalaureate program in biomanufacturing. So the growth has been unbelievable, in my opinion. We grow so fast in just this is our fourth year in just 3 years. So in terms of the enrollment also going strong. So here, as I mentioned, we started off our first grant in 2021 September, and in the first year 2021-2022 we built the program, and then we opened our first door in fall 2022 with the first cohort, and ever since then we experienced growth in the enrollment. We continue add the section, including two sections of dual enrollment into different high school. But from there, we hit bumpy road. It's not all the sunshine and rainbow. When we start entering high school, we encounter a lot of some issue, and this is where we realize two things. Number one, if we have a champion in that high school, we have the someone who can advocate and make things happen. Things will go smoothly, right? So things will happen essentially, right? Starting with student recruitment and talking to administration and so on and so forth. If you don't have that champion in the high school, things would be very difficult. It's still possible, but it's very difficult. That's one lesson we learned. The lesson number two we learned is that well, actually, I know I'm quite oblivious, as a community college professor, quite oblivious of the high school system, and vice versa. The high school staff also perhaps not quite familiar with the community college system. So there's that miscommunication that oftentimes create barrier for this building a pathway from high school to biotech training at community college. So we thought, hey, what a better way to build this, to raise the champion and build more communication than to apply for another project and using this ATA project to make that happen. And that's basically what we did. So we applied for this second project, which we're gonna talk about how that play a role in here. And basically, just to continue on, this year we are on track to beat our expectation from last year. And again, this based on the enrollment in the fall 2025 and the current enrollment in spring 2026, which as of right now, it's full enrollment plus I think 8 or 9 people in the waiting list as of today. So and then next year we're gonna start another two section of dual enrollment, and we anticipate to serve over 100 students over 100 new students every year. So that's sort of the expectation moving into 2026-2027 academic year. So now we're going to the project, which is Illumina Genomic Discovery. What is Illumina Genomic Discovery, right? Well, this is a classroom curriculum that was created by Illumina, the company Illumina Inc. in San Diego, and with the collaboration with Generations team and the Biocom California Institute. They built the classroom curriculum to learn about their platform, the technology, which is the sequencing by synthesis technology. They draw upon the real case scenario when there's a food poisoning, food contamination with this enterohemorrhagic E. coli or Shiga toxin producing E. coli, STEC. And basically through that activity, students learn how to prepare the sample, prepare library, and then clean up, and then doing sequencing and actually performing data analysis. Now, I provide this QR code for you if you want to get access to some online genomic resources, videos, or virtual tour or something. It's available over there. You can scan the QR code. It will direct you to the Illumina website or Generations team website, and then there's a lot of videos over there that you can use for your class. Now, as a part of this, they have this so-called lab in a box where they prepare the box that has all the laboratory equipment, supplies. The teacher come and check out the box for several weeks, so they can bring it to their high school classroom, teach their student about the next gen sequencing, and then return the box, right? So the distribution is this actually already existing in San Diego. So when I first heard about this, I thought, hey, can we bring this to Pierce College? So I started asking around, and they said, well, it may be difficult because LA, Los Angeles is outside of our service area. And we thought, hey, well, in my opinion, if we can introduce next generation DNA sequencing technology in the classroom, that would be wonderful to bring excitement for students and also bridging the gap between students who are thinking about career in medicine into biotech through what we call precision medicine. Right? This is the foundation of this whole thing. So I thought, is it possible to expand it to Los Angeles? There's only one problem though, right? Me have to go to San Diego to pick up resources, right? Which is fine. I'm okay with that. But then it would be unlikely for every single high school teacher to drive to San Diego because there's no distribution center in Los Angeles. So we started discussion, hey, what if we create distribution center in Los Angeles through maybe, funding from NSF? And oh, let's chat. And that's basically where the collaboration start. So anyway, so that's basically how we get started. And then basically in this project, we have three goals that one is to empower high school teacher and college partner educator to become community leader that will be able to show student the path to go into biotech industry, right? The second and the third is actually somewhat similar. Second focuses on building the distribution center, and the third one is to build to train biotech workforce through authentic student research experience in genomics. And the first couple we partner up with three high school partner, and then the hope is for each of these high school to have a path through dual enrollment to biotechnology training program, not only at Pierce but also to other uh, uh, community college in their service area. In fact, one of them, Huntington Park, is not within our service area. They will be connected to another college, I think LA Trade Tech or East LA College. We also have additional community college partner, our neighbor LA Mission College and El Camino also will join. They have joined for our virtual training and will join our in-person training next Saturday at Pierce. So if you have met us, you probably have seen the presentation from our student Mastura, who actually make this instrument work and use it as a part of her project at Pierce. And basically this box that you see over here is what the teacher will check out. So we will have this Pelican case with pipettes and all this other stuff, the qubit and holders, centrifuge, mini PCR, things like that. And then we will have our student interns building upon our internship model to prepare this so that the teacher can check them out. And just my last slide over here, if you also attend the ATEPI conference in Washington, D.C. last year, 2025, you may have talked to another student, Fatima, who started this project by using, learning not only the Illumina genomic discovery pathway, but also using it to look at the gut microbiome of a horse. And that has inspired another student who was in the path to go into veterinary school, and he's with us today, Khalil Robinson, to expand upon her project to do this in the large mammals. So and that's without further ado, I'm going to let him talk, and he actually end up joining our biotech program, and he will be joining our biomanufacturing baccalaureate cohort in this year. So without further ado, here we go. I'm going to share his slide. Khalil, are you ready to present?
I am ready.
All right, take it away.
So,
Wait, let
hello.
me.
Oh, go ahead, go
I
ahead.
got the swap. There you go. Yeah, there you go.
All right. So, hello, everybody. My name is Khalil Robinson. Like Dr. Kamajaya was saying, I'm part of the third sect of what we're doing with this process on teaching students that are looking to go into biotech to help build up a repertoire of students who have advancements and understanding within the industry as a whole through like Aspire and other programs. So going through the process of our project that we did, which is an expansion of a previous student, like he was saying, which was Fatima, is that we were looking at identifying microbes within fecal samples from four different species we have on campus access to at Pierce College. As you can see on our first slide, that would be horses, sheep, goats, and cattle. Go ahead, next slide. Perfect. So just to give you guys a little bit of background, especially for introducing myself, I'm a student who started out in LACCD community colleges. I started off with Los Angeles City College and transferred to Southwest College to kind of figure my route on what I was going to do to get to a university. I ended up transferring to Cal Poly Humboldt State, all the way at the tip of Northern California. And I was currently doing a post-baccalaureate work at Los Angeles Pierce College and ended up finding the Aspire program, which helped push my interest and understanding within biotechnology. So one of the main things that was important to me was asking this question on like, looking at significant potential applications on why we would want to look at identifying these microbes and what use could we do learn from them. And with being within vet med, one of the biggest things I learned was that it was an area and also an industry that is very underlooked and underserved. We don't really know too much about their microbiome, and then also what potential uses could be within the industry, within another industry. So that was one of the significances I was looking at for this project. So the main research goal was to characterize and compare the gut microbiome across multiple livestock species and using NGS sequencing or next generation sequencing and generate exploratory data to identify these microbial patterns. And that way that it could be something that can be used for future antimicrobial discovery and downstream screening uses that using a modified tiny earth protocol. So that was the initial timeline of our project. One of the good things that we do within this class that we were doing for biotech is making sure that we're able to use timelines and understand our roles, kind of pre-preparing us for working within the industry. And this was just a general workflow of what we did. So for my project specifically, I actually had to do the field work and go out and not only get permission from our other departments, but also go and interact with the animals and get the samples myself. It was dirty work, but it was fun. And then next, you develop the library prep and then set up the cluster generation so that way we're able to sequence what we're looking for within our animal samples and then use the analysis from Illumina. So it was a very long and arduous process, but it was very worth it and very fun. So just I don't know if you guys are up to date on animal science, but just to give you guys a quick anatomy rundown, between us as humans who are monogastric, which is usually a singular stomach and singular pathway for eating, with the animals that we were researching, they're either a ruminant or a hindgut fermenter. Pseudoruminant is another species of animal that we could look at, but we don't have that on campus. So the cows and the sheep and the goat were ruminants versus the horses who are a hindgut fermenter, which means that they don't ruminate in the same efficiency as the large herbivores, but it's close enough that we're able to get a good level of variance, but similarity at the same time. So I just want to show you guys a little pictures of our samples and collection and field work. The two gentlemen that you see on the right are our leaders at our school for our pre-vet and our equine science department. So, one of the good things about this project is you have to learn how to communicate, which you do within biotechnology. And it was an important aspect to not only get permission, but also make sure that I'm doing this process safely and following IUCAC standards. And just a couple more photos of me and all of the animal subjects because we like to give them their props. Thank you guys for your poop. And then here was what we use for our extraction, our library preparation. So because these animals usually either ingest or are around soil-borne pathogens, we were able to use the D&Easy Power Soil Pro Kit for DNA extraction, and then we were using the aluminum ISEC 100 for our data acquisition. We were able to, within the Spire program, we have access to specific lab equipment that allowed us to do identify and also perform quality control. And these were some of the equipments that I used during this whole process to make sure that my process was working correctly. And with that, we were able to get our results and look at our data, which NanoDrop was helping us QA/QC it. Our gel reading that we did for PCR showed that we had proper DNA bands to be able to run our samples through the ISEC 100. And then, as you see on the last slide, our last picture, that we were able to fully sequence all of our animal samples that we collected. And voila, this is what we received at the end of all of that. We received our multiple corona charts. As you can see, we have a negative control just so that way we can base what we're looking for in comparison to all of the other animal samples. So in my process, since I was comparing it between, I needed to identify what species of bacteria that we are seeing within the animal samples. So I compared it to topsoil and organic soil layers within the school area, just so that way that I was able to see if I was going to go to the next process, which is to develop antibiotic processes. Um, I wanted to make sure that what I'm seeing in the soil is similar to what we're seeing in the animal's guts. So if you go to the next slide, Dr. K, and go ahead, just tap on each one of those. You can click the other one too at the same time. So what we're able to find out is that looking for a very common species, Lactobacillus, I was able to identify that yes, we do have Lactobacillus not only in our soil samples, and you click on to the next slide. You can click to the next slide. And then you can click on all of those. Oop, go go back.
Oh, fast. Okay, this one.
Yeah,
Okay.
you can click on all four of them at the same time. Between our ovine, our bovine, our equine, and our caprine, we were able to find this same Lactobacillus species, letting us know that not only did our processes work when isolating and extracting our DNA, but that means that once we do our next process, which is to isolate those specific species for testing for the potential antibiotic properties, we know that it's there. So between our whole corona chart, which we're still going through to identify species that are both within the soil and within the animals, we know that we're doing what we're supposed to do correctly. Though there were some hiccups, one of the best things that I learned within Aspire is about GLP, which is good laboratory practices. And between us and another student who was actually a high school student who was working in the same vein as what I was doing, we ended up having issues with communication and identifying for our specific samples and how we were supposed to key them. And so what happened is that our sample got read wrong, but luckily, with our process and our SOPs that we're following, we were able to still get a sample readout for her to be able to do her project, which was on lactobacillus within yogurt. So the next steps and future direction of this project is to then kind of do another QA QC check with it by running a nanopore for the same idea to make sure that we can compare between what Illumina is doing and our nanopore, see if everything's lining up perfectly. So that way that the following step is when we isolate and culture our individual species that we want to check for antibiotic process or antibiotic properties that we know for sure what we have, and for future references or reproductions of the project, that students will be able to follow and know which animals that we have on campus that have these specific species of bacteria that we need to utilize for study. And just with closing remarks, I want to say this project was very, very long. It was very hard, especially because I was doing it by myself. But it was one of the greatest joys I've ever had with working with science, especially in the undergraduate level. Without this opportunity, I don't think I would have been able to do it, especially at a community college level. And I am happily and appreciative and blessed that the Aspire program was allowed had the access to the materials and the time and opportunity to allow it for us. These are acknowledgments. So not only the Aspire program, but Dr. K, Fatima, who was um, the vanguard for starting the project, where she did it originally with equine, and a couple of other individuals who were essential for the project's success. And then references for you guys if you wanted to follow up or look through on some of the things that we use, ideas and material that we helped utilize to get our process going and the finishing up with our project. And a wonderful thank you guys for listening. I really, really appreciate it.
Thank you.
Thank you. That was a great presentation, Aron and Khalil. Our second Project Talk speaker is Dr. Oana Martin. She is joined by her colleague, Dr. Mary Ellen Kraus. This project strengthens Wisconsin's bioscience talent pipeline by expanding biotechnology career awareness and dual credit opportunities. Thank you for joining us.
Hello. I think I'm sharing the right slides. My name is Oana Martin. My colleague Mary Ellen Kraus is on this call. We appreciate the opportunity to share our progress in the project STEM to Biotech Careers in Wisconsin. This is our first year of the grant. We are about 6.5 months in, I would like to tell you very briefly about the goals and then launch into a couple of the activities that we've done so far and some of our preliminary results that will guide our future activities. So my co-PIs here, in addition to Mary Ellen, are Dr. Kit Carlson, and Kit is a biology faculty at Madison College, and Alexis Kruger, who is a faculty in the electromechanical technology program. So we have a cross collaboration here across between departments and between two different schools that are meant to enhance exposure of students to biotechnology careers. We developed 3 goals, and I will mainly talk about the first goal today in terms of activities. That's the one we launched in first because we have a baseline, some more experience working with high schools that we're building on. We started working on the second goal and the third goal, but I will save that for next year's presentation. So our first goal has to do with high schools, and we focus on expanding awareness of careers, career pathways, supporting our teachers, both our dual credit teachers and other science teachers at our partner schools. The second goal has to do with this enhancing our collaboration between different departments at our college because we feel the biotech program has a unique situation at our school. It's separated from the other STEM programs. We are in a different school. So we felt the need to increase our visibility to the students in these schools and create materials and career exposures for them. And the third goal has to do with researching the factors that affect the retention of students who are in the STEM university transfer pathway. That's how it's called at our school. Students who come to Madison College with the intent to transfer to a 4-year university and pursue a STEM career. So we've noticed that there is quite low retention in this pathway, and we are really interested together with our co-PIs in the other departments to understand the factors and hope to redirect some of these students and increase their interest in biotechnology. So I will focus more on the first goal of our grant, maybe touch on the second goal with one of the activities today. So far, during our first year, we've done quite a few things to work with high schools. We worked on curriculum and toolkit development, and we have so far 2 modular kits for high schools that I'll briefly talk about, and started to include pathway mapping. So one of the unique things about our kits is that they include career and educational informational materials that are specific to our college, our area, and Wisconsin. The second point here, we focused on creating and perfecting, I should say, our student immersion lab experiences that we conduct in our labs. So I'll mention some of those. We are hosting students in our biotech labs and doing immersive lab experiences, and we were trying to see how well that's going, what we can do to increase their interest in biotechnology. And Third, we've onboarded some teacher mentors for our teacher mentorship network that we're trying to create with our dual credit teachers and new science teachers, and increased our interaction with our local industry partners in terms of this collaboration and this project. So to give you a little backstory of these kits, we have a dual credit program, a quite vibrant dual credit program here that's been created by my colleague and my co-PI, Mary Ellen Krause, over the last few years, and several dual credit courses in partnership with high schools. But we've started looking at data, and I'm showing here data from numbers from 2019, so right before COVID. So the numbers are make a little more sense. So out of the 109 students enrolled in biotech dual credit, we know that 41% of them enroll at our college and 65% enroll in college, but only 6% enroll in our biotech program. So these are our dual credit courses. So we were a bit puzzled by this, and we decided to address this discrepancy by tackling two of our higher enrollment courses. These are biotech applications and biotech career seminar. These are one credit courses that are taught in the high schools by high school teachers. And we decided to expand the materials we offer, the support we offer to these teachers by creating some hands-on kits. And here are the two that are the most advanced here that we finalized and piloted a little bit with a few schools. The first module focuses on enzyme and the food biotechnology industry. And it highlights enzymes as biotech products and careers in the food industry. So students perform hands-on experiments to explore enzymes, and they learn how to micropipette. They work with catalase, and we highlight careers in this area, including some biomanufacturing careers as well, and tie this into the cheese industry in Wisconsin. For the second module, plant medicine, this focuses on the pharmaceutical industry, developing pharmaceuticals. And here as well, students do some hands-on experiments. Again, micropipetting is involved, and then I will actually elaborate on some of these experiments with examples, but they are all related to the process of drug development and careers in this area. To give you an idea what's in these kits for the first module, so simple equipment that the teachers can use and supplies that can be replenished, for example, dry yeast, hydrogen peroxide, things like that for consumables. We do provide this, as well as enzyme, chymosin, that they have multiple experiments. An example of a lesson and materials, students learn how to micropipette in detail. They practice, but we try to gamify this a little bit. So they do a pipetting challenge, and then the student with the smallest percent error can win the challenge, for example. Another example would be a lesson centered around chymosin that ties concepts to recombinant enzymes, recombinant proteins, the cheese industry. So students will compare the curdling of milk at different temperatures, and then we talk about the mechanism. For the second module, we simulate a Kirby-Bauer diffusion test with acid-base chemistry. So we provide all the supplies, and that way the students can learn, in addition to concepts of drug development, they learn hands-on, for example, how to do this kind of testing. So what's unique about these kits is that they connect skills to careers, and careers that are specific to our industry in Wisconsin. So we have for example, a career match game. That's a classroom activity where students match these career cards with the jobs with the skills required for each job. So by looking at these, they could tie these skills to concepts and skills that they actually learn during the class. So we believe that they gain more confidence in pursuing biotechnology careers and perhaps seeing themselves in these roles as future scientists. We are also creating some roadmaps that connect educational pathways with career pathways. And again, these are adapted for our state and for our companies here. So it's showing students that they can start with a foundational degree, like an associate's degree, even a technical diploma, and enter our industry. And as they advance their education to a bachelor's degree, they develop more skills, they advance their career pathway as well as a scientist. Perhaps then if they pursue a higher degree, they can get to a leadership level in this industry. So we're trying to offer them a long-term pathway and motivation to pursue this sort of careers. So the second activity that I want to mention here is our lab immersion experiences for high school students. These are dual credit high school students, and they come to our biotechnology labs on our campus for extended experiences. We develop these 3-hour experiences where they do PCR and spectrophotometry, even cell culturing with CHO cells. We also include a career pathway presentation. And we started looking at, so we've been doing this for a while, but right now we started looking at the impact that they have. So I wanted to show some survey results, and I realized the numbers may be a little bit small, but what we noticed here is that we're really impacting their career awareness here. So they come in with only about 16% awareness of what the broad biotech career means, and then they leave at 87%. So that's the highest gain we're seeing. We're also noticing a significant increase in their lab skills confidence. And we are thinking of confidence as retention in the biotech pathway, because if they're confident about their skills, they can maybe see themselves in these types of careers. We're also increasing by 29% their interest in pursuing a biotech-related educational or any type of experience. So interest in this pathway by offering information. So one last thing is this experience is related to the second goal. It's also a lab experience that we've done this with students from Madison College that are in this STEM Academy pathway. So these students are they are in high school, but they're taking classes at our college as part of the STEM school, the STEM Academy. So they came in for this. This was a shorter 75-minute lab experience. And they're already committed to STEM because they are in this STEM pathway. So about 88% of them plan to go to a 4-year university, but only less than 20% named biotechnology as their pathway interest. So biology and engineering dominate here. So we feel that this is very important information for us as we move forward in trying to create clarification about this pathway and materials to provide information. Students do believe strongly in the impact of science according to their responses, but they are unclear how to become a scientist. So we believe that we can close this gap moving forward by creating more clear pathway materials and increasing the exposure we offer students in these pathways, in general STEM pathways, to biotechnology. So we're very excited for the future. We're looking at a few different things for the rest of the year and into our second year of the grant. We are launching some summer biotech boot camps at some of the branch campuses that Madison College has to increase our reach into the rural Wisconsin areas. We are planning a summer teacher workshop, Biopathways Wisconsin, this coming summer to expand our network of teachers and industry mentors. And then we are really starting to look more at our outreach and recruiting data. So using data to assess the awareness gap and inform the materials that we are focusing on creating. So I would like to thank you very much for your attention. Mary Ellen and I have our email here, and we are happy to answer questions. Thank you.
Thank you for your presentation, Oana. And at this time, we have about 14 minutes for a Q&A to any of our presenters today. You can either raise your hand or we can also go to the chat if there are any unanswered questions.
Well, I was curious about your curriculum. Can we get your curriculum for the some of the outreach you're doing in Madison?
Oh, sorry. Yes, yes. So you mean the kits or the in-house curriculum for
Yes.
the immersion?
Yeah.
The kits? Yes,
Oh,
yes.
maybe yeah.
We've just finalized them. So I think we're in the process of scaling and building this to send out to more teachers. And I'm trying to figure out where to post this. Yes. I would love to have opportunities to share.
Oana, I'd be happy to help you with posting it.
Thank you.
You guys have contributed so much to the course in the box section. I mean, all the lab math and Lisa's stuff with basic biotech skills, and your course with chromatography skills. Yeah, Mary Ellen with lab math. I said that. But yeah, it'd be wonderful to add this too.
Great.
Khalil, may I ask a weird question that isn't what you were studying and you might not know the answer? But I know you were looking at bacteria. Did you happen to also identify archaea, and do you have any idea, like percentage or something like that? Sorry, I know that's weird. I just care about archaea sometimes.
So we're still going through the data that we collected. I think I did see some archaea bacteria within it, but like I said with the corona charts, we're still searching through it because it's a lot of bacterial species, so I have to personally go through it one by one. But when we do find out, I would be happy to let you know.
Yeah, there's people who care about methanogens, and you'll have some, and actually, it I doubt it'll tell you anything, but some of your organisms should have them and maybe some of them shouldn't, but I don't think that makes sense. Pay no attention to me. But thank you. Cool work.
Yeah, I
Thank
think
you very much.
Yeah, I think the I mean Illumina platform, they have this base platform which is very good. It gives you this really beautiful corona charts you can easily look at. But there's some limitation, right? Sometimes you get a hit that's not being mapped onto their database, and then it just say unassigned or something like that. And we have, we're still working on trying to using data analytics, scale to extract that data and then perhaps do some more computational outside of that base platform. So
Well, Sandy, you have a question.
Not a question actually, so much as a request. Both of you. Oh, I'm going to lower my hand here. I know both of Aron and Oana. You're both working with high schools, and I'm not sure. One thing you may not realize is that we don't necessarily see those high schools is connected to your programs unless you get those high schools to join Innovate Bio. I don't think any of the ones you work with, Oana, have joined. And Aron, I'm not sure about your high school partners, but please encourage them to join Innovate Bio, and they'll be part of the network. They'll show up on the map, and they'll show up on your program page, and they'll help us track the different colleges that have dual credit, dual enrollment programs.
Good idea.
Yeah.
Will do. Will do.
Yeah, and
Thank you.
if any of you were on the last presentation where I was presenting, the federal government is very interested in high schools' connections to colleges. So it really helps Innovate Bio to support you by getting them listed in our website associated with programs.
And it showcases the work you're doing. So,
Yep, exactly. That really helps us. Then I can expand to how many number of high schools we're working with. So, it's interesting to me. So I have a dual credit program, as you know, that Joseph runs. And plus now we're getting more teachers involved. So we're adding on 4 more high schools. Yeah, the number of students, they mainly go to the 4-year school at this point. But that's all right. I'm going to capture them now in the 4-year school with my advanced technical certificate, because then they articulate back. And Aron's going to capture them probably more because with a 4-year degree, that bias to a 4-year degree is, but it is hard. Yeah. I mean, it's like, even if you are a 2-year student, you will run into a ceiling, even in industry, where you'd have to go after your 4-year degrees. So, but you just have to provide them with various on-ramps and off-ramps to do it.
Thank you for mentioning that, Linnea. I always wonder if this is a unique situation that we have with the 4-year school mentality. But yeah, there are options, though. We've noticed with our alumni that they complete online bachelor's degrees. So after they have all these hands-on skills, they start building a career. It is easier to get an online bachelor's in not even, sometimes it's not even science, it's more management, and they build after that. So I wonder, probably you're seeing something similar in
Texas.
Yeah.
So initially it started out where the community colleges would do the industry-based skills, and then the next 2 years would be management. So then they would articulate with management programs. I do think, I'm sure, Aron, you've adopted the 4-year program, correct, that everyone is doing in California, Miracosta, and
Yep.
very
We
similar.
standardize our curriculum with the other 4 biomanufacturing program in the state. Yes.
So we pretty much told anyone in Texas, if you do that, you're going to adopt California's 4-year program. But it is interesting now. Our community college offers 4 4-year programs, but the state of Texas limits how many 4-year programs you can do. And I bet for many states, it first starts in health science. And Cassandra has a cat. I wonder what the microbiome of the cat is. I have to, I know I'm
that's
asking.
online.
That's online. I'm really curious about hindgut fermentation with the horse. I had no idea. So I'm going to have to look this all up now.
Real quick, one of the things that I want to say was it was a really good thing, Dr. K, and bringing me to not only this talk but also other ones, because it's really interesting how there's not a lot of people who work within animal science who also like kind of cross respect into biotech. So whenever I go to these talks, I know so much about animal science because I'm an RVT and I'm looking to be a vet. And so when I tell people this, for me, it's like my group is common knowledge, but then it's like everybody's like, what? There's a huge differences between it. But yeah, horses, they like in the project that we did, what was really difficult is that between the two, horses are actually really inefficient as fermenters. So when they eat stuff, it comes right out. They're almost like us, they're being monogastric. So we had a harder time. We actually sometimes have to go into the horse to actually get the samples to make them as pure as possible. I know it's gross, but we do it for the love of science. But yeah, it was a lot of variances. The main thing though is that because they all were herbivores, we did see a lot of similarities, and that we're seeing more and more similarities as we're going through the data. So it's really cool.
So I hope the group doesn't mind. So Sandy knows this because we did a Chautauqua course in Alaska. And when I was in Alaska, I got to meet the wildlife biologist who had the moose herd. And the reason he had a moose herd is that he was figuring out for the state of Alaska what moose eat. So he would airlift these moose all over the place with a helicopter and watch them eat, and they eat willows. And then he wanted to know what was going on gastrically and isolate the bacteria. So one of his moose had a little pouch connected to his, I don't know, whatever you call it, stomach to get the bacteria out. But I want to tell you, having a moose pet, these things are big. And they all ran up to you to say hi. So your work is really applicable to a lot of different areas. So wildlife conservation is one of them.
Yeah. And one of the main, like, far future goals is to possibly develop an industry within the industries you already have, like within dairy, meat, textiles. So, like, as they're processing the animal, they could also use them for like possible probiotic and gut microbiome development. So that way, it's like we're not adding more to the population, overpopulation issues of what we already deal with animal products, but just utilizing what we already have and able to like garner resources from it without adding to it. So like that's far future thought plan of it, but that was like my
Well,
goal.
I know there's a market for probiotics for cats and dogs. People will buy their pets anything. Go
Yeah.
for it. Fantastic
I'm
job.
unconvinced, Linnea, that I should write a grant about airlifting moose, though, no matter how hard you pitch it. I'm a little worried for the safety of the students involved. Aren't we all looking for more enrollment and not like moose deaths on campus?
Yeah. But you know what he proved to finish the story? We have a minute. He proved the the governor of Alaska wanted to kill more wolves, saying it was the wolves that was doing in. But listen, what it really was, they figured out it was really about willow, the availability of willows, because moose will have two calves, but unless if they have to have enough food, enough willows to get it. And the state of Alaska was doing improper burning of willows. They weren't allowing for a greater burn for healthier willows. So it wasn't the wolf. So he saved the wolf by proving that it was really the state of the willows and that the state of Alaska has to do a better job of burns.
We see that same issue with deer on the East Coast because they eat all the low shrubbery, which removes like hiding places and also like biodiversity. So like when they took all those cornerstone species like wolves or like predators who are attack like their like equine centers or their cattle centers, it caused like a huge downstreaming issue. So like yeah, no, I 100% I agree. He was a great job for saving the wolves because they need them up there. It also prevents the moose from overeating the willows, which would cause a similar problem, just a lot
Yeah,
slower.
yeah. And don't forget, there's more moose in Anchorage than there is in the wild because the moose have figured out if you're going to have babies, have it around humans so wolves and bears can't get you. There you go.
The polar bears don't care though.
No, no, they eat humans. They would if they could.
So at this time, I'd like to conclude our project talks. It's 1:30, and I don't want to take the rest of your time. Thank you to our project talk presenters and guests today, and then to all of you for joining us. We will post the recordings on the Innovate Bio website by the end of next week, and then you can find out more on these two featured project talks as well as future project talks on the Innovate Bio website. And as Sandy mentioned earlier, please join Innovate Bio. It is free, and you will have access to our monthly newsletter. Again, have a wonderful weekend, and we'll see you next week.
And stay warm.
Yep.